Excellent. We start in a minute. Okay. Hello, everyone. Hello. So let's start. Thank you. Thank you very much. Thank you all for coming. So our speaker today is Daniel Ruiz Renes. Most of you know him. He did a PhD here under my supervision. It was already in 2019. Was that? And after that, he's been a postdoc in KU Leuven, working in the group of Linder Gelens. And while he's now back here, now he has a contract with the Momentum, the SIG Momentum, okay, working on Posidonia patterns and other things. And Daniel today is going to explain us, or going to tell us about what he did in his postdoc in the lab of Linder about spatial heterogeneity accelerates phase-to trigger wave transitions in frog egg extracts. Okay, so thank you very much, Neil.
Thank you, Daniel. Thanks for the nice introduction. As he said, I'm going to talk about this work, spatial heterogeneity accelerates phase-to trigger wave transition in frog extracts. Is a work that recently we published in Nature Communications, and one that I feel quite proud of it, or that I enjoyed doing, and it's part of the work I did in the lab of Laboratory of Dynamics in Biological Systems, led by Linder Gelens, many of you know. So I thought that since I did a PhD here, and many of you know, it would be good to say a few words about what I've been doing apart from this work that I want to present today. So I prepared just 2 slides just to show you a bit the different works I've participated in this during my postdoc. So essentially the lab of Linder Gelens is part of the Department of Molecular Medicine at KU Leuven, of Cellular Molecular Medicine at KU Leuven. And it's a big department with many groups specialized in different topics. And the group of Linder Gelens investigates mostly, topics related to experimental biology, data-driven modeling, and computational physics. And my role in that lab was mathematical modeling. The lab of Linder Gelens does experimental biology and try to investigate processes that affect the cell cycle, reconstituting in vitro the cell cycle. And for that, they use the animal model Chinopus laevis, and more recently they incorporated zebrafish too. And they use here you can see an egg of these frogs and how it divides with time, right? So that's one of the animal models they use, or the experimental setups they have. But they also can use cell-free extracts, which is a very powerful technique to reconstitute cell cycle processes in vitro. So essentially what you do with these eggs is you centrifuge them, you crush them, and you put them in tubes, and you can, for example, reconstitute here the formation of nuclei, but you can reconstitute also the formation of the cytoskeleton, which is formed by fibers of microtubules. So here it's like a space-time plot. This would be time, and there are different snapshots. And in green, you can see the formation of nuclei. So essentially, they take the cytoplasm of the cells, they put them in these tubes, and the different structures that constitute generate the division of the cell, reconstitute themselves in a self-organized manner. So I'll explain a bit more in detail the experimental setup of the main publication I want to explain today. but I have collected some of the publications I participated in during these years, and mainly we studied theoretical aspects, like for example, in this work with Pedro Parra, which many of you know, we investigate, for example, the biochemical autonomous oscillator that drives the cell cycle. What happens, for example, if you incorporate different regulatory networks that give rise to what we call different bistable switches. Or we also investigated, for example, did modeling on the self-organization of the cytoskeleton of the cells. That's the work I'm going to present today. This is about mitotic waves. And we did also a more theoretical paper, a review on the Fitz-Nagumo. That's because the models we use are typical. They typically have relaxation-like oscillations, and we use a lot the Fitz-Nagumo. So we did a review on that with Daniel Cebrián Lacasa, which did here. And we also have more experimental results, analyzing, for example, spiral waves of these microtubules, the organization of the microtubules here in yellow, and also we have experimental results of this cell-free extract in droplets in oil. So they made a microemulsion of extract in oil. So they can reconstitute artificial cells, and they can analyze the division, the cell cycle period by looking at the nuclear envelope breakdown. So that's the typical things we do there, and the words that I have. But today I want to focus on mitotic waves. I'll give more details about that. So first I want to introduce you a bit like the main concepts, because now have been like a lot of information of different things. So essentially when we have a cell like the frog egg of the Chinipus laevis, you have the cell division cycle. So the cell divides, and there is a biochemical autonomous oscillator that tells the cell when to divide. When the activity is high, the cell divides. This has been shown or characterized using cell-free extract, which is what I already introduced, this experimental setup they use in the lab. How you do that? Well, you take you inject hormones in the frog, they lay eggs, and you collect it in the morning. And essentially you apply, well, you would need to add some buffers, but essentially you centrifuge them, and you have different steps of centrifugation, and you totally crush the eggs and essentially obtain this cytoplasmatic fraction, which is the content of the cell. And with that, you can do many kinds of smearing. For example, analyzing the activity of the main cell cycle regulator, which is cyclin BCDK1, or you can reconstitute or recreate the formation of microtubules, which are the fibers of the cytoskeleton, the formation of nuclei. Many things have been done using this set. The biochemical oscillator that has been characterized using this animal model of these cell-free extracts has the following phase. So essentially, you have the synthesis phase, the interphase, in which you have synthesis of the main cell cycle regulator, cyclin B CDK1. And it builds up cyclin until one threshold, the activation threshold. And at this time, essentially, the activity of cyclin BCDK1 goes very rapidly up. This moment here indicates the cell when it needs to divide. And the high activity of this complex triggers many mitotic events. For example, the depolymerization of the cytoskeleton, microtubules disappear or disassemble, you have nuclear envelope breakdown and other processes. For example, it also triggers the activation of the anaphase promoting complex, which targets cyclin for degradation. And when you have this activity, degradation is higher. So cyclin starts to degrade, and you reach a second threshold, and the activity goes down. And with that, you have a periodic oscillation of in the activity of the complex cyclic BCDK1, which essentially is the clock for the cell, right? So 30 minutes. Yeah, you can make it longer, for example, if you dilute the extract or different perturbations, but typically least in the X 30 minutes. What? I mean, you can activate this process without fertilizing, but you need to add calcium ionophore, which is a way of activating, I mean, of mimicking what the sperm does. But essentially, when you have the sperm, you do the same or similar things. I think you can also start it with electrical shock. Right? There are different ways to do it. So what happens if you put this autonomous oscillator in long tubes of 150 micrometers? Yeah, and a millimeter, 10 millimeters long. Yeah, exactly. Well, then what you start to see is that you have in red is high activity. You have periodic oscillations that are more or less synchronous in time and start to desynchronize with time and emit waves of high activity, which trigger different events.
By activity, you mean concentration?
No, by activity, I mean the conformation of the complex of cyclic BCDK1. This complex has different sites. And they need to be phosphorylated. So this means adding a phosphate group. So when you have the right conformational state, then we say it is active. You measure this activity with a friend sensor. That's a novel technique, at least for Chinopus laevis. It was developed for somatic cells before and for Drosophila few years ago by the group of Stefano Italia. And essentially what this Fred sensor does is designed such that when it's closed, both the yellow and the blue emit light at the specific wavelength. And when it's open, they emit at a different wavelength. So, and this closes and opens depending on if there is phosphorylation and dephosphorylation activity. So this tells you if your complex is phosphorylating. Yeah. Yeah, activity is having this. Essentially, in our models, what we look is the total concentration and the concentration that is active, to simplify things. Okay, so I was saying that it's a novel technique because these experimental results were new. I mean, we didn't have like a direct visualization of mitotic waves using this sensor before. We had it in Drosophila, which is a half a millimeter in size, and doesn't allow you to observe in such a way the presence of mitotic waves like you see here. So this is quite suitable to analyze the spatiotemporal dynamics of the waves you observe this biochemical oscillator. No, no, this is extract in a Teflon tube and it's immersed in oil. So essentially, it can be slightly affected by the bond, but it's not much. We don't see like relevant effects from. Okay. Well. So these the results of this work were the result of a collaboration with the University of Michigan, with a group of Kyung Yang. And they did the experiments as a part of the PhD of Owen Pools. And they weren't very sure how to analyze properly the data. They did in a manual way, let's say. And we started this collaboration to analyze it better and also to provide some mathematical modeling. They, as you can see, they do similar experiments as they do in the lab of Leonard Flens. So what we did is to take this data and make space-time plots, or kymograph as they call it, and we identify the different waves that you can see here. And essentially with that information, you can measure locally the speed and the period for the positions of the wave and local measures. And you can also measure, for example, the activation rate at a specific position. And with that, you can do a more systematic. For example, one of the things you can see is that at the beginning you have synchronous oscillations that start to desynchronize, and you start to have phase waves, which start to become trigger waves, and at the end, it's dominated by trigger waves. No? And it's something that you can see here. Phase waves are like a result of desynchronization of the oscillatory of the oscillation, let's say. So all the activity grows, let's say, homogeneously. And at the end, you have more like traveling pulses that propagate. Yes, just a different way of visualizing. So with these local measurements, what you can do essentially is to repeat it for all the experiments and obtain the time evolution of the period, and we calculate the inverse of the speed, which is the slope, because it's very flat at the beginning, and you have a divergence here. So these values are quite small. So, but you can easily calculate the speed by getting a smooth curve with locally weighted smoothing of the data that you see with the density plot, which corresponds to the extracted data. And with that, this one? Yeah.
Spatial.
No, the period in time. So essentially you measure the period, computed from this, the difference in time of these two oscillations, and you associate it to that position. And you have it for this time, and yeah, you can repeat it for all the times, let's say, all the times that you have oscillations. Not for all the positions. So, well, you can extract the dependence, and you essentially observe that the speed goes very rapidly down, and you have a later slower relaxation that we can quantify with an exponential fit and determining where the exponential fit starting to, let's say, fade. That will be, this kind of measurement will be useful at the end to compare the results. And also another thing we were looking for, when we plotted the wave speed as a function of period, you can see that the period in extracts naturally increases from 50 to 150, and essentially in an almost linear way. So essentially, you can interpret also period as time, right? So the things moves like this, evolving in time. So we were interested in the relation of wave and period because in previous results experiments of mitotic waves in Drosophila, it was proposed that the change in period that you observe typically in Xenopus laevis extracts would modify the properties of the oscillator and as a result affect the propagation of the waves. And that's one thing we wanted to check. So for that, we investigate if this was the reason for the change in speed or was due to the time needed for a wave to stabilize to the trigger wave, which is the stable in comparison with the phase wave. We did mathematical modeling to discriminate between these two cases. So essentially what we did is to take an existing model that phenomenological in the sense that it only accounts for a couple of mechanisms that are the most relevant for describing the cell cycle instead of describing very complex regulatory network, but it's parameterized with experimental measurements. So and essentially describe different processes. So here you have the total cycling and the cycling that is active. So essentially both are synthesized at a certain rate and it rapidly binds to CDK1 forming the complex, and the activity of this complex is regulated by these two feedback loops, which are described by these functions. And essentially you have positive double feedback loop, as they call it, with CDK25 and a negative double feedback loop with Q1. So, and you have the most important, which produces the oscillations, is the negative feedback loop between APC. So cycling, the activity of cycling with CDK1 triggers the activation of APCC, of the anaphase promoter complex, and this targets cycling for degradation and creates the oscillations. But just for completeness, I included like the response that you have for this function, but this is something that's experimentally given. So I think it's more useful maybe to look at the null planes of the system and the phase plane, which you have here. So essentially during interface, when cycling is synthesized, so it moves to the right, it reaches the threshold of activation. And in the active state, you activate the degradation and it goes down until the second threshold. And this gives you essentially a relaxation like oscillations, which you can see here. So the system, if you change synthesis, that's like the next video. So you are moving this curve if you change synthesis, and here you have the bifurcation diagram. For low synthesis, the only possible state is the inactive state. For other values, it's the active state. So these are fixed points. And in between, you have oscillation. You have a window of oscillations that in synthesis that gives you oscillations for the. So our goal here was to try to mimic what we see in experiments and try to discriminate. And what we did is to introduce different scalings of the parameters to get the dependence we see in experiments of the period. So essentially, we can control different processes. You can control the activation deactivation rate, or the synthesis degradation with the different parameters that you can see here in the corners, or you can do a global scaling. Essentially, what we see from experiments is that the activation rate does not depend, it's constant in time. So then we discarded this regime and we attributed to the elongation of the period and scaling like this. Right? So essentially, the elongation of the period that you see is because the synthesis changes with time and you have a longer interface duration. So we implemented that in the model. That's what you can see here, the dependence of the parameter that you should obtain from the experiments to mimic the period, and you can see here the period changes, and we included spatial heterogeneity to force the formation of. And what we immediately saw is that essentially increasing the heterogeneity, you can control the relation between wave speed and period. So here you have different values increasing the strength of the heterogeneity that we can put in space, and in that line you have the experimental couple. So in a way you can decouple the wave speed, you can decouple it from the period. So essentially it's not the change that you observe in experiments due to of the wave speed, it's not to the change in period. It's just due to the transient effect of trigger waves. They need a certain time to entrain the system. So initially start synchronously, and a trigger wave needs some time to entrain the surroundings, and it's what you see here. So essentially at the beginning you have phase waves, and it goes to a fixed value of the trigger wave.
So you distinguish between the waves or traveling waves and the trigger wave. Or the oscillations, we are talking about homogeneous oscillations or oscillations with a bound wavelength, and the trigger wave, which is a pulse. Is that the difference?
I mean,
to go from the oscillations to a trigger wave, what does that mean?
So you start with homogeneous oscillations, right? And you end up with a pulse. So since you start with this initial condition, you need some time for a trigger wave to dominate the system.
But why do you go to that? Because the are unstable? Because of heterogeneity?
No, the homogeneous oscillations are stable. No, are stable. But since you have spatial heterogeneity, there are differences in period due to the heterogeneity. You introduce difference in period. Yeah, synthesis is not homogeneous in space. Synthesis is the control parameter that we put in which we introduce heterogeneity, and these regions that are faster, oscillate faster, essentially need some time to entrain the system.
Okay, so when you have it's defecting, so the place where it oscillates faster is the one that
later coordinates the propagation of waves. Yeah.
So this it's an heterogeneity of
this is the amplitude of this profile. Let's say. To simplify. Yeah, so that's the synthesis in space that is different in space. That's the way we introduce heterogeneity. Okay, and essentially, well, that's a measure of the amplitude of this profile. So the bigger the heterogeneity, the bigger the changes in synthesis that you introduce, but the faster it gets entrained.
One of the beta.
Yeah, that's what we get from experimental results. We know that changes that way. So, and later we introduce the spatial heterogeneity that it is in a natural thing of the experiments, and we observe that the dependence that was claimed between the period and the speed, it's not because of the properties of the oscillator are changing, but because just the transient time that you need for a region that is faster oscillating to coordinate all the space and train it, let's say.
Are you saying that this will be stabilized? That the period will be stabilized? Because there we don't see that. It looks like it's going to die out, the oscillation, because the period is longer, and since it stopped the simulation or even the
Well,
experiment, what do you
here
expect?
we stop the simulation. In experiments, this elongation of the period, there is no explanation that it's verified with experiments. The current hypothesis is that you have ATP depletion, and essentially this controls synthesis or other processes related to synthesis. So essentially for the experiments, what you start to see here, I don't know if it's in the previous time or if we already have it. No, but you will see it in the next chimeras I'll show. We call it the dies out, because essentially you start to have oscillations after some, well, typically they have 15 oscillations or it's a transient state. Yeah. So we do not account for this in the modeling, the fact that you lose the oscillations. In the modeling, yeah, this will continue increasing. It's a simplification.
The fastest oscillations are the ones that are driving the system.
Huh?
Why the period is increasing? It should not to be. As the driver, as a driving effect, you should be able to have everyone fast. Sorry.
Well, yeah, it does. It reduces the period. No, I mean, it will have the period of the guy that is fastest, but all of them are changing. All the periods are increasing in time because the global synthesis, let's say, it's going down. Well, that this part was to try to discriminate if the change in speed was due to the change in period as originally proposed, or as we show, was due to the transient effect. And it's essentially this change in period is due to a transient effect. And if you change the period using mathematical modeling, essentially the change in speed that you would observe, it's minimal. I mean, it's 50 or less. So you cannot explain with mathematical modeling a big change in the wave speed, and it only can be explained by this transient effect of starting from synchronous oscillations to a regime which is driven by the fastest, and you have a trigger wave. So what we did next is to explore further this experimentally, and we added sperm chromatin. This essentially, when you add it to the extract, makes the nuclei to spontaneously form. So in here you can see the same waves, but you will see there are like some difficult to catch, some circular spots that correspond to nuclei that are formed. Right? So if we look at again at the chimegraphs, as I was saying, you can see here, for example, a nucleus triggering a wave, but also there are other nucleus that are the nuclear breakdown happens because of the wave comes and passes, let's say, and increase the activity. So what you can see immediately is that when you have nuclei, the entrainment goes faster. So you transition much faster from phase waves or synchronous oscillation, which you almost don't have at the beginning, to a well-entrained system, which you have one that is faster and coordinating big regions of space. Related to your question, this is what happens typically, that after some time. So if you can look at the same things, we can identify from the very beginning more related to pulses, although not super visible, but you can see that at later times. And we can compare the case without nuclei in green and the case with nuclei. And you can see an immediate growth of the inverse of the speed, the slope, which goes to a fixed value, which is the value of the trigger wave, the stable. So in here you can see the same plot for the two cases. respectively with the colors. And interestingly here, what you can see experimentally is that you can decouple the period from the speed. So essentially this tells you that it's just a transient effect. It's not the fact that the period, well, the period or the oscillator properties fix the value of the speed, at least when it's relaxed. No? At the later stage, it might change a bit the speed due to the change of the oscillator, but that's not something easy to identify or characterize. Okay, so later just we did bit of modeling to try to illustrate how this works when you have nuclei. And what we did is to incorporate these terms to the modeling, to the model, which are essentially import terms according to a function, a Gaussian profile, very simple, to mimic the structure of the nucleus and the cytoskeleton. Essentially, you have in the cytoskeleton molecular motors that move cargo and import it to the nucleus. So you have a typical size for these structures that give us the size of this shape, and we can include an import term following this profile according to the gradient of this function, and that gives us the flow. Yeah.
This is an attractive potential.
Yeah.
Okay.
Yeah, this is
The-
attracting cycling to the center. So essentially what you can see here is that if you include that, cyclin accumulates at the position of the nucleus and is depleted at the surroundings because of this import term, and this helps to activate at this particular position of the nucleus faster than without it, right? And here you have the formation of the wave. And if you look at the phase plane for these two positions, well, you see that it crosses the threshold earlier. Very surprising. What we did is to incorporate different nuclei, and with incorporating different Gaussians. And what we do is we include this Gaussian profile, but we put a temporal dependence, let's say, well, not a temporal. We activate import when you have low activity of cyclin lucidity-1. When the activity is high, essentially you have nuclear envelope breakdown. There is no nucleus, no microtubules anymore. There is no import. So you will switch on and off depending on the levels of cyclin, the import term. And this import term is given by a Gaussian, and we can choose these parameters according to the experimental values and put some noise on the values of the parameters or sample it from a Gaussian distribution. And we can do simulations with multiple nuclei. So here in black, you see the levels of cyclin, and in red, you see the activity. And you have the activity here and the levels of cyclin. And the dots correspond to the different nuclei, and the strength is indicated by the size and the color. So they don't move, we fix the positions, and you can see how they emit waves and start to interact. So this, rather than having like a quantitative result, it was more to illustrate how we can train the system faster. You don't have a direct way to characterize like the amplitude of the spatial perturbation you put. So in this sense, you cannot directly relate it to experiments. And interestingly, it gives you, well, it shows how different nuclei could interact by sending waves and inhibit each other depending on the distance they are. Last thing we did is to apply an external perturbation to the extract, and for that we wanted to verify which extent the spatial heterogeneity triggers the entrainment of the system. And we added what we call cytostatic factor. It's essentially extract that is prepared in the active state. And it's arrested, so it doesn't cycle. and you dip it with a tube like 5 seconds, and you remove it, and essentially you can trigger waves in this sense. So first, what we did is to prepare an extract that is in the low activity state, arrested in the low activity state, what we call interface extract. And there you can see the formation of a pulse, and we can measure the speed, which agrees very well with the speed that we have in the previous results, which is the speed of the trigger wave, that's stable, let's say, speed of the wave. Here you can see what happens if we put this perturbation in an extract that is cycling, as the ones I've shown before. And essentially in this case, you have the perturbation, it's triggering waves that are emitted to the other direction. And you can do the same with nuclei, adding spare chromatin, and you see a similar behavior. And well, here you can see the nuclei form. So here you can see the different chymographs, and you can see how this active extract diffuses in, and you, without nuclei, you have almost synchronous oscillation, but the entrainment goes very fast from the beginning. And if you have nuclei from the very beginning, as we already know, you have the formation of some waves that it's completely passed by the formation, by the waves that come from the bond. So here, essentially, it's the same plot again, but what we can see is that it goes very rapidly to the basic level. So essentially, we are showing that applying these perturbations is a way of entraining the system very rapidly. And finally, what we did is to compare the different entrainment times, or the time. We have two measurements: the time in which we do an exponential fit of the tail, and we have the time of initiation of this exponential fit. So essentially, if you take the green curve, you can do exponential fit here, and that's where the exponential fit is good enough. So we quantify these times and the length scale of this exponential, I mean the temporal scale of this exponential fit, which are summarized here, and essentially show what I've been explaining, that including nuclei entrain the system much faster, and and also applying the perturbations from the boundary, it's a strong effect. Interestingly, when you have nuclei entrain combined with the perturbation, you have a cumulative effect there, so there are no big surprises. And we were able also to quantify, according to, well, in agreement with previous results, the speed of mitotic waves. So with that, I would like to finish. I would like to thank my collaborators, especially Lenz and the group of Pyongyang, here especially Owen Pauls, which is the PhD student who did the experiments, mainly he did the experiments, but also the rest who participated in preparing figures and helping with the experiments and so on. And thank you for your attention. And you have
Well, thank you very much, Daniel. It's time for questions. First, Roberta.
A curiosity, you were showing the model with this H function that you told are extracted from the experiments. Are these linear, non-linear, time-dependent, time-independent?
time-dependent.
Time-independent?
Depends on the concentration.
So for instance, H, this H of A?
So this controls the activation and deactivation of the of cycling.
But for instance, the first stage is linear in A or non-linear? This is the behavior of this function.
Yeah, as a function of the activity. So Sorry.
Thank you, thank you.
Essentially, this would tell you that
I-
this term for low activity is high, no? And
No, thank you. I
it
missed
decreases the
the,
activity, and you have for higher activities, this increases the activity. Essentially, this gives you this folded shape that we call a bistable switch. For these values, the activation is difficult, let's say. It's prohibited, and for higher values, it's enhanced.
and then another very basic question. At the beginning, you were telling that you were actually destroying the eggs. So the clock behavior is something that is in each fundamental component you're telling.
It's in the cytoplasm. So in the cytoplasm, you have pools of cyclin. I mean, you have maternal mRNA that produces cyclin, and you have like a pool of that. And so I was
No, it was just, I was thinking, you are actually looking
yeah,
at the clocks of the fundamental component, that when you put them together in a cell and they are not destroyed, they
it's
still
not,
maintain.
you need to add some buffers like to avoid the proteasome, so the proteins that degrade other proteins in the cytoplasm. You need to prevent that. But essentially, you crush the egg and it's still functioning. And it's not only functioning like the biochemical oscillator, but the cytoskeleton. So there are like papers in which the cytoskeleton is able to divide like a cell would do, just in these extracts where you have like destroyed everything.
Cytoskeleton is actually what divides them.
Yeah.
Breaks into the cytoskeleton without that it is in the liquid in the
Yes, in the
extract.
extract.
Doesn't break anything because there is no
Exactly. So in fact, in interface extract, it forms like a pattern. It's mostly due to the interactions between the cytoskeletons, or like a Voronoi diagram.
example when I see.
Yeah.
Okay. I have three questions. But one, why does the thing start oscillating homogeneously? It's because you start with a well mixed liquid,
Yeah, it's mixed, and you keep it on ice.
Oh.
So essentially, when you remove it from ice, it starts to oscillate.
And because it was well mixed at the beginning. The
Well,
reason
yeah.
why it's homogeneous.
It's well mixed in the sense that you centrifuge them, and yeah. Yeah, there are works of the lab, and there's like a window of in which you have oscillations. Yeah, a temperature window in which you have oscillations. So you arrest them with temperature.
Okay, then in the periods you show are of the order of 1 hour or more, but you say that the period was 30 minutes. Is this the difference between in vitro and in vivo, or what is the?
Yeah, it can be a difference. In the egg, it's 30 minutes. But if you put it in extract, depends when it's quite variable, and it can depend quite a lot, for example, on dilution. When you prepare the extract, you put the eggs in a tube, and you have to remove all the water that you can to like, yeah, you need to be precise on that. And it's not something that you control, so it might change because of that or other reasons. I mean, you have also the variability of not all frogs are the same, not all eggs are the same.
Yeah. And final, you talk about nuclei, but those are not the cell nuclei. I mean, are there cell nuclei? They have some membrane, and they have some. Is this really forming?
Yeah, they have. I don't know which picture is the best to show this. Well, here I don't know if I can see.
So I only seen the microtube, but the real they have some lipids on it. I mean, they have some proteins on the border.
In the cell device, it breaks.
So, for example, these are experimental results of the lab done with droplets of extract in oil, embedded in oil. You vortex it, and essentially you get this mix of droplets. These green spots, they are nuclei. The way to visualize that is you add GFP-NLS, which is green fluorescent protein and nuclear localization signal. And essentially this a protein that is fluorescent that goes into the nucleus, and you can observe it like that.
Is this just,
And it has
because I
membrane.
perhaps I missed that you are adding DNA, so the chromatin, or what are you?
You add sperm chromatin.
Okay, so then this
Yeah, you
I
can-
didn't go that.
You can add also sperm like directly to no, but we did that to trigger the development of the frog.
Okay.
The entire
So there is some DNA, so it's maybe a real nucleus. Okay.
It's something you can quantify, and in this work, I haven't done it, but they quantified the amount of DNA they have and how influences the changing period.
So, thank you very much. I want to make a follow-up question to the difference between the system that you have, which is the cytoplasm plus nucleus, but you don't have cells anymore because you have broken them apart. Comparing that with the cells that you have in the eggs, like in nature. So, how relevant, for example, are the patterns that you see, and in the end, the trigger waves, to what you would see, in eggs in nature? Do you see these kind of waves?
I mean, it's difficult to find like a direct application of why it's useful. To me, the most relevant conclusion that you can take from working with extracts is to better characterize the biochemical oscillator. There are like, I mean, it may be seen that you know a lot of things, you know how to model it, but there are many things that we still don't understand, and that's a way of like doing things in order and verifying. But typically, what is said is that in early development for the eggs, this is a millimeter in size, so it's quite big. For other organisms, it's not so relevant, but for big organisms, you need a coordination of the whole cytoplasm to trigger the division of the cell in a synchronous way for all the egg. So this, in this case, these trigger waves are said to be relevant because with diffusion alone, you wouldn't be able to synchronize all the entry into mitosis.
I see. And it's just a curiosity, but when you do this experiment, do the nuclei also double? Like, for example, in the experiment, in the mathematical one that you did, that you have your nucleus here, I saw that they didn't change after that happened, but shouldn't be two afterwards.
You don't have like a perfect division as you would have in a cell. Sometimes you can see it. Sometimes, in fact, we see like divisions that give you three. So that's why yes and no, because it's not so perfect, but sometimes it happens. Yeah. In fact, in the you can see here, for example, you have like different dots, let's say different circles, and that are like different compartments, but we haven't analyzed them very precisely.
Okay. Allow me one final question from my side then. Do you have the shape of this trigger pulse more or less characterized somewhere in the? So it has this square shape like the relaxation oscillations, or?
I mean, looking at the oscillators, and now I'll show you the pulse.
Yeah, exactly.
This here.
A little bit. Okay.
I don't know what to say in the sense if that's due to experimental. I mean, if it has this shape due to the experimental.
Yeah, it's a bit top flat here. Yeah. Okay.
So,
okay.
I mean, this, as I was saying, is a way of directly measuring the activity, right? But it's a bit sensitive in the sense that you need to you have two wavelengths of emission in the fluorescence, and you need to do the ratio between the two to get the curve. But there are always
All right. All right. Thank you very much. Okay, so there are no more comments. Thanks a lot, Daniel.
Es también media hora como en la división celular.
Empieza en 50 y acaba en 150. Un poco más largo.
Un poquito más largo.
Sí.
O sea, los building blocks triturados de los huevos son un poco más
Sí.
lentos que no donde están los
Sí. Y si le pones agua y está más diluido,